is going to teach us about Lynch syndrome. So, Lynch syndrome is a genetic condition in which an individual will have an extremely increased risk of developing cancer, specifically colorectal and endometrial cancer, in their lifetime. By talking briefly about what cancer is. So, cancer is the uncontrolled growth of cells. So, there are two main genes that are of interest in cancer that are oncogenes and tumor suppressor genes. Now, oncogenes are mutated forms of proto-oncogenes, which they drive and signal cell division. So, under normal conditions, they signal the cells divide when they're supposed to, but when they mutate to become oncogenes, that signal is always on, and it continually sends that signal to divide. And tumor suppressor genes, they serve as checkpoints in the cell cycle. They serve to stop cell growth and division if certain conditions aren't met, such as insufficient DNA replication. So here, this figure to the right shows the hypothetical accumulation of these tumor suppressor and oncogene mutations to go from a normal somatic cell to a cancer cell. Shows us what happens in cancer development in a colon. Initially, you have the colon serosa that starts mutating and growing uncontrollably. Eventually, you get to what's called metastasis, where those cells now leave the tissue of interest and spreads to other parts of the body. And these images show endoscopy images of what cancer looks like in inside of a person. Cancer a little bit, let's talk about what Lynch syndrome is. So, Lynch syndrome is a hereditary condition in which the risk of developing cancer, specifically colorectal and endometrial cancer, is extremely increased. It is inherited in an autosomal dominant manner. This means that you only need one mutated copy from either your mother or your father to inherit the condition, and also means 50% of your offspring will have this condition. This is caused by deleterious mutations to any of the genes involved in the DNA mismatch repair. And we'll talk more about that in the next slide. But the DNA mismatch repair is the spell check of DNA replication. And these genes are classified as tumor suppressor genes. So, Lynch syndrome accounts for around 2 to 4% of all colorectal cancer cases, so kind of small, but individuals with Lynch syndrome can have up to an 80% risk of developing cancer. So, it substantially increases cancer risk in these individuals. So, mismatch repair system, or MMR system, its function is to serve as the spell check of DNA replication. How functions is you have four protein subunits that come together to form a complex that locates mismatch bases and repairs them. It's encoded for by the protein subunits are encoded for by MLH1, MSH2, MSH6, and PMS2 genes. If any of these genes acquires a deleterious mutation so that one of these subunits is no longer able to function properly, this whole complex no longer works. And so as a result, these mismatched bases do not get repaired by this complex, and the accumulation of mutations begins to happen at a much higher rate in an individual with deficient mismatch repair system. Consequences of the accumulation of mismatched bases is you get what's called strand slippage. This occurs when the mismatched bases will still attempt to line up with its complement DNA strand, but as a result, you get this bubbling going on. So this results in either an insertion or a deletion mutation. So the amount of insertion deletion mutations increases much more substantially when you have deficient mismatch repair system. The strand slippage, we get large insertion deletion mutations in repetitive regions of the genome. For instance, we have what are called microsatellite repeat regions, also known as short tandem repeats, all throughout our genome. The number of short tandem repeats or microsatellite repeats should remain constant in every cell of the body. However, a tumor that has microsatellite instability has number of repeats that are different in tumor cells compared to the rest of the body. This indicates that there is high genomic instability, and it is indicative of a deficient mismatch repair system. This genomic instability gives rise to a much higher mutation rate and also creates much more chromosomal abnormalities, which are very common in cancer development. Detect if an individual has mutations to any of the MMR genes. And so an individual with Lynch syndrome, which is the deleterious mutation to any of these MMR genes, they can regularly get screened for colon cancer through a colonoscopy procedure. It is recommended that a Lynch syndrome patient receives a colonoscopy once every 2 years after they turn 18 years old. So regular screening can catch early cancer development before it even becomes a problem, before it becomes malignant, and this significantly increases the chances of survival and also a fully healthy recovery. Briefly about the current research and advancements that have occurred in the last few years, there is a vaccine for Lynch syndrome that is currently in development. So mismatch repair deficient cells, they actually generate high amounts of a tumor-specific neoantigen known as neoAGs. So this vaccine, what it aims to do is it delivers these neoAGs to the patient through injection, and it trains the T cells in our body to target cancer cells that express these neoantigens, so that once these cancer cells start developing, the T cells are able to take care of them much more early on in the process. Presentation. If you'd like to learn more information, I'd strongly suggest you to go to the CDC website where they talk about Lynch syndrome. Understanding this disease and knowing more about it can help save countless number of lives. And thank you again for joining me on this video guide of Lynch syndrome.